Hi, everyone. It's Miss Kopczynski. And today I'm going to be talking more about your chapter 3 notes. And Buster's here with me. So if you hear something in the background, it's probably Buster because he's kind of noisy. But no matter, we are going to pick up where we left off and finish up some notes. So I'm going to share my screen with you here and get the slideshow going. Oh, so many things open. Okay, so we left off with talking about the plasma membrane. I'm going to skip ahead to where we were. So discussing the types of passive transport. So we talked about sledding down a hill and how this represents the concentration gradient. So I kind of went over this slide in class. And the next type of transport that we're going to discuss is active transport. And this is going to move substance from that area of low concentration to higher concentration. So this is going up the hill. It requires energy. So it's that ATP is that form of energy, and it requires membrane protein transporter because we need, it has to have energy. So here's an example of an active transport using ATP. So energy again derives from the breakdown of ATP, and it's used to change the shape of the carrier protein. So again, it's almost like a lock and key. So it's gonna the protein's gonna come in and it will let that transport know, hey, you need to open up so we can allow more of that nutrient into the cell. For example, water is a really easy one. So if it needs to allow more water into the cell or water out of the cell. Here's co-transport. So some carrier proteins use energy from the downhill transport of one molecule to transport another. So sometimes they need a buddy to go with them. And that's kind of what I think about. So if you're like, nope, I don't want to go it alone. This is just like these proteins. They don't like to go it alone either. So they will pair up so that they can make it through. That's that co-transport. I do have a few videos in the learning log that are put on our Monday date. So if you want to take a peek at those, because I did put a couple extra videos out there so that you can look at those and go back and watch them. They are from Bozeman Science, or if you want to rewatch the videos here that I'm creating for you, wonderful. As always, I'll give in-class examples. Please ask questions. Okay, endocytosis and exocytosis, that's going to move materials in bulk. So I gave the example of my Play-Doh example in third hour. If you were in second hour, I drew it on the board for you. But that endocytosis is going to bring things into the cell, and that's why the prefix is endo. So it's moving into the cell. So it's going to butt up against that cell. The cell's going to recognize it and it's going to slowly pinch it off right into the cell. And exocytosis is going to be the complete opposite. So here's that picture. Again, it's hitting the outside of the cell, and then it's going to start pinching it off to form that vesicle. When it's exocytosis, it's going to go in the reverse way, and then it will release it outside of the cell. It's like, goodbye. Okay, so I do have some videos of endocytosis and exocytosis as well. Again, I have those in the learning log. So if you want to take a peek at them, or if you need me to provide any more examples, I would love to do so for you. So information transfers across the plasma membrane. So receptor proteins span membranes required for transmission of information to and from the cell. So receptor sites interact specifically with those signal molecules. So they're going to signal each other. And a change is triggered within the cell as a result of being a signal molecule to that receptor site. So once that signal comes, it's going to respond to that. Okay, so different cell types have different receptor proteins. Each one has its own different receptor, just like we have our own response to things. So here's a receptor site. So once that receptor goes in, and it's going to cause a reaction. So there's a substrate and then the product. And then the receptor, that receptor will leave. Okay, here's the big meat and bones, the sodium potassium pump. So I know a lot of you know a lot about this already. And it helps maintain our cell volume. So sodium comes in, potassium comes out. And if you don't know, it's 3 sodium ions for every 2 potassium ions brought into the cell. So again, it keeps those unwanted ions. It keeps, if it needs one, and it maintains that cell volume. It's going to increase in cell volume and increase in water in the cytoplasm by decreasing pumping and allowing more sodium inside the cell. It will decrease in cell volume, and then this is when less water in the is in the cytoplasm, but it's increasing pumping and expelling more sodium ions. So it can work both ways. So here's a picture of it happening. So again, you're starting with step 1 here, and it's going to bring the sodium ions are going to bind into that binding site, and only from the cytoplasm. So it's going to go into that binding site, the binding site of 3 cytoplasmic sodiums to the sodium potassium pump, and it's going to stimulate that breakdown of ATP. It's going to then release that ATP, causing the proteins to change its shape, expelling the sodium ions. So it's not going to release them until that protein changes its shape, and then it's going to release them. The loss of those sodium exposes 2 binding sites for potassium. So that's what happens. Once those sodiums leave, potassium is going to come in, and that's going to trigger a change so that it can come into the cell. Most of the potassiums will diffuse out of the cell, but sodium diffuses in only very slowly. And sometimes you can have leaky gates, which I'm not going to get into. So toxicity, this is relative to concentration of solutes into fluids. So if our pump is not working right, we can have we can get a toxic cell, and then cell death will occur. Isotonic, that's when the extracellular and intracellular ionic concentrations are equal. Cells will maintain a normal volume in isotonic extracellular fluid, and then that regulatory mechanism maintains extracellular fluid that is isotonic with intracellular fluid. Okay. So again, this is helping maintain that cell volume. Okay. So there's a variation in toxicity. So it can be either hyper or hypo. So hyper is when it's higher than the intercellular fluid. So that's when the extracellular fluid is higher. So the outside is higher than the inside. So then water will diffuse out of the cell, and then the cell will shrink and die. If it's hypo, that means that the extracellular ionic concentration is lower than the intercellular. So that means inside the cell, there is a higher concentration. So water will diffuse into the cell, and the cell may swell and burst. So here are some of the organelles that you'll see in the cell. So some basics here, the nucleus, that's a big one. And that's going to be that information site for DNA. The mitochondria, mitochondria, the powerhouse of the cell. Again, that's where that energy is produced. Lysosomes, that's going to clean everything up. You got our plasma membrane, so it's allowing things in and out of the cell. Our ribosomes are that site of protein. We have our vesicles, so they are it's like a shipping container getting waste out of our cell, and then also bringing things into our cell. Our Golgi apparatus, it refines and packages those macromolecule products, and it can bring them to places that, or ship them to places elsewhere in the cell, as well as shipping them outside the cell. Rough endoplasmic reticulum, primary site of protein synthesis by ribosomes. Then we have our smooth endoplasmic reticulum. This is the primary site for our macromolecules, our exoskeleton. So this gives us our shape and structure to the cell. We have our centrioles, which are our microtubule structures, and they are involved in that cell division. Peroxisomes destroys that cellular toxic waste. Okay, and then cytosol is that gel material inside of the cell to hold those organelles in. Okay, anabolism. This requires enzymes. This is part of the metabolism that we have in our body. So anabolism is building things up. So it's used in making and assembling large molecules. Anabolism is building, so it's making molecules. And it does require energy. Anything that's going to build needs to require energy. And it's, again, assembling those cell components, and it's going to, it's used in storing energy. Catabolism is the other type of metabolism, and this is going to break down. So I always think of C, it's cutting, it's breaking down things. And this requires enzymes, and it's going to release energy. So it's breaking down nutrients and then recycling those cell components out. So it's used to access energy storage. All right, glucose provides the cell with energy. Glucose provides energy for the cell and it generates ATP. So in the absence of glucose, there's other carbohydrates, fats, and proteins that can catabolize, so break things down so that we can generate more energy. So here's that process of things getting broken down. So even in cellular, we have our cellular respiration and that cellular work here. So that cellular work is going to be the anabolism, transport, so that we can get muscle contractions. And this energy is required to create this ATP. So there's 4 different stages of cellular respiration. We have glycolysis, preparatory step in our citric acid cycle, and electron transport system. Okay, so here are those steps broken down. And again, you need energy for glycolysis to happen, and you do get ATP as a result. It can also be go over to that electron transport system, and then again, that's a result of ATP is a result of that as well. Okay, so summary of energy production from glucose. There's over 20 enzymes that catalyze reactions. There's approximately 36 ATP produced from each molecule of glucose. Oxygen consumed, carbon dioxide produced. Pretty easy. So oxygen's consumed and carbon dioxide is produced from that. So cellular respiration is that process that uses oxygen and produces carbon dioxide in that process of making ATP. So it's part of that byproduct. Okay, I'm going to stop here because we are going to cover some of this. We're going to cover this in class a little bit more and break it down into smaller bite-sized pieces. So we'll talk about the citric acid cycle, this glycolysis that's happening, as well as that electron transport chain, and then hit on that preparatory step a little bit as well. Thank you for tuning in. And I hope that you guys have a wonderful weekend. And I hope you all have fun at the game. Go Panthers.